Okay, now, in order to be able to talk about ganglion, sensory neurons, and things of that sort, we have to talk about neural crest cells because those play a key role in forming the ganglion that are going to synapse with the spinal column and with the brain at key locations during development. Now, though they originate from the ectoderm, as I mentioned before, they have a multipotency, almost pluripotency, depending upon who you ask, to become cells that not only you would expect to come from the ectoderm, such as neural tissue, but also things that you see in other germ layers, such as bone and cartilage and renal cells that are part of the kidneys and things of that sort. So there's a wide variety of tissues that they play a role. They become muscle in the heart. Their potential is huge. So the biggest question becomes, how does this happen, and why are these only in vertebrates and not in invertebrates? This is a very big evolutionary question as well. So where do neural crest cells originate from? They originate at the point of junction where the ectoderm and the neural tube meet. So during the primary neurulation process, the junction where you get this folding right here, once you get the infolding and the ectoderm seals itself off and the neural tube forms, there's a group of cells that form just between those two. That's where the neural crest forms, all along the axis of the embryo. So there are actually 4 different groups of neural crest cells that we're going to talk about today that originate in different parts, some in the head, some near the heart region, some in the trunk region, some that will contribute to the parasympathetic and the sympathetic nervous system. So I'm going to give you a brief overview of those, of the peripheral nervous system as well. So here's where the neural crest cells originate from. As you get closure of the neural tube, these cells receive various inductive signals from the overlying ectoderm as well as from the neural tube that will give them their respective fates. So here is an example of an embryo showing you the 4 major regions where the neural crest cells come from. One of the regions we call the cranial neural crest cells because they form in the head region. Then you have the cardiac neural crest cells. These are a group of cells that only form right here where the heart's going to form. Then there's the vagal and sacral neural crest cells. These form at opposite ends where you get the parasympathetic neurons starting to form. And then you have the trunk neural crest cells, which are going to form all along this area here. So they are overlapping. So when we talk about these various regions, they're not discrete regions. There is overlap between these various regions. But we find that there are several clusters of the neural crest cells that originate from these regions at various times that form the vagal and the sacral neural crest, or the cardiac neural crest, or the trunk neural crest. The cranial neural crest is really one of the few that's exclusive to the head, doesn't really have any overlap with any of the other neural crest cells. And that actually plays a key role. One of the things we're going to see today is that cranial neural crest cells have a higher potential to become a wide variety of cell fates than those in these trunk regions. One of the things being that the trunk neural crest cells do not have the potential to become bone or cartilage, whereas the cranial neural crest cells do. So there is a restriction that is applied to the neural crest cells in this region, primarily due to the Hux gene, as we'll go over. Okay, so here are the huge set of derivatives that the neural crest cells can become. I mean, I'm not even going to go, we're going to go through most of these today, but this is just kind of a list. You've got peripheral nervous system, you've got endocrine, you've got pigment or melanocyte cells, you've got bone and cartilage, connective tissue, muscle, all sorts of different types of cells. So it's a huge endeavor to understand how the neural crest cells form and what they contribute to and what are the mechanisms, the developmental mechanisms of that. Now, in order to talk about some of these, I have to mention a little bit about mesoderm right now. What you're going to see today is these tiny little spheres that are adjacent to the neural tube. We haven't talked about the mesoderm yet, but you do need to know one part of it, and these are what are called somites. Somites are these balls of mesoderm tissue that form every hour and a half in chick development. It takes longer in others, but in chick development, we use them to actually measure developmental progress. And they're just these large spheres of mesoderm that essentially become the vertebrae of the spinal column, as well as some tendons and other parts. We're going to go into all the derivatives of the somites later on. But the somites, as you'll see, play a key role because they influence the neural crest cell migration and how the cells migrate to their respective locations. So that's why I have to mention them now. Is these they almost look like hemisphere, little teardrops right here. They initially start out as spheres and they start morphing as they go through their gastrulation events. So that's what you're seeing. These little dots, these are somites. So you're going to see me talk about, hey, between somites 1 and 3, you have the cardiac neural crest. That's just so we can reference where these cells are coming from. They're still coming from the junction between the ectoderm and the neural tube, but we can say in this region between somites 1 and 3, the cells that migrate from that's cardiac neural crest. So just so you know, somites are these spheres of mesoderm that form all along the axis or the trunk of the embryo that will play their respective roles. And we'll talk about those when we get to mesoderm, but you are going to see a couple of things that they influence today. So here again, you're seeing these spheres. In fact, you're only seeing half of the spheres of these somites. So here's one of the things about neural crest cells is depending upon where they're found, they have various migration patterns. And these migration patterns depend a lot upon what's the surrounding tissue and what influences they receive. The somites play a key role in the trunk and the cardiac and the vagal and sacral neural crest migration, but there are no somites in the head. So in fact, the head, the migration of neural crest cells in the cranium actually have more to do with other regions that aren't somites. They're what we call rhombomeres. So the rhombomeres, or what I told you, the rhombencephalon, which is the hindbrain, these play a key role. So we're going to see that some of the same influences in the rhombomeres take place as in the somites. So let's start with the trunk neural crest cells. There are two main migratory paths of these neural crest cells when they first form. They can either migrate ventrally. So remember, they form in the dorsal region between the neural tube and the overlying ectoderm. And when they migrate ventrally. So they'll come around the neural tube and they'll start coming downward through the somites. Okay? There are some situations where they migrate between somites, but it's not really important to talk about those right now. We're just going to look at the main migratory paths. So the main migratory paths are either through the somites in what we call a ventral lateral migration, or we have what's called a dorsal lateral migration. Usually dorsal lateral, which means that it migrates more laterally in the dorsal region of the ectoderm, so kind of around the somites on the outer rim rather than through the somites. These are usually late migrating neural crest cells. So early migrating neural crest cells tend to go through the somites. Late migrating neural crest cells tend to go around the somites. Some of the genes that we have seen in the somites forming are things like FOXD3, PAX3, snail, I believe in slug. These are just some of the developmental genes that are responsible and necessary for the formation of the neural crest cells. They're finding more and more as they go through, and other key differences between some of the neural crest cells in the head as well as in the other regions. The signaling that starts forming the neural crest cells typically comes from the neural tube as well as from the ectoderm. So you kind of get this combination of signaling from the overlying ectoderm and from the neural tube that will then cause genes to be turned on right at that junction that form the neural crest cells all along the anterior-posterior axis. Now, there are other factors coming into play here in genes that will influence whether or not they're cardiac neural crest cells or vagal or sacral or trunk neural crest cells and so on and so forth. So we're going to go through each one of those in turn. Now, what's fascinating, and you're going to see this for both sets, is that the migration of these cells, they have to go from an epithelial to mesenchyme transition. So they have to break free from their epithelial layer, and they have to then become these free cells that will migrate independently and move through these other cells. To do so, there has to be a lot of cytoskeleton arrangement and changes in that. But what's interesting about their migration is that even though cells will migrate through the somites, they only migrate through the anterior region of the somites. The primary reason for this, so if you look at a cross-section, here's the neural tube, here's the notochord. The neural crest cells are actually fluorescently stained here, so you can actually see these migrating ventrolaterally through the somites. Well, if we look at the posterior somites, look, there's no migration whatsoever. These cells will not migrate through the posterior region. So the only way in which they can migrate is through the anterior somites. So if you look at a somite as if it's a sphere, there's an anterior half and there's a posterior half. The anterior half, that allows somites or allows neural crest cells to migrate through. So what's one of the key factors that allows them to do that? It's the relationship between an ephrin protein or a ligand and an ephrin receptor. Now, the somites, the anterior portion of the somites are releasing this ephrin protein. In fact, usually when we say the ligand, we use a lowercase. So when you see ephrin in your book with a lowercase e, that's the ligand. When you see a uppercase E, that's the receptor. I don't know why they do that, but they do that. So, so the ephrin receptor that's on the neural crest cells, what you'll see is they'll actually be able to migrate through the anterior region. But here in the posterior region, you don't get any type of migration because when the ephrin ligand and the ephrin receptor combine with one another, it causes the cells to stop migrating. It prevents them from actually migrating any further. So where there is no ephrin ligand being secreted, the neural crest cells can actually migrate through because there's no ephrin ligand receptor combination. This is also true when there are motor neurons, which we're going to talk about a little bit later on today. When the motor neurons start projecting their axons out from the neural tube, this is also the same mechanism, is that you get this repulsion from the ephrin ligand and the ephrin receptor so that the axons cannot migrate through the posterior region of the somite. They can only migrate through the anterior region of the somite. So not only do neural crest cells have the ability to migrate because of that, but also the axons or the growth cones, as the axons start being projected out of the neural tube, the synapse with muscle and other tissues and whatnot are also restricted to this. So in the trunk, the somites, which are mesoderm, play a critical role in influencing the migratory patterns of both neural crest cells and the axons as they're being formed. Now, here's just some examples of a couple of the genes that influence. As the neural crest cells migrate along the various lengths of the embryo, certain neural crest cells are exposed to certain genes. Some are exposed to Wnt signals, other exposed to BMPs, other exposed to endothelin and glucocortical hormones. The exposure of these multipotent neural crest cells is ultimately what allows them to take on their fate. So as they're migrating out from this region and they migrate through the various paths, they are influenced by paracrine signaling by the adjacent tissues. And depending upon where they end up will determine whether or not they become, adrenal glands cells that are part of the on top of the kidney, whether they become Schwann cells, which are going to myelinate many of the axons that are going to be projecting from the neural tube, whether they become sensory neurons themselves. So a lot of the Schwann cells, which are cells that myelinate or surround the axons of the neurons that are generated by the neural tube, originate from neural crest cells. And some of the sensory neurons themselves that form the ganglia are also coming from the neural crest cells. Some melanocytes, which are the skin pigmentation cells that you have in your skin that generate melanin, and that's what causes your skin to darken as you are exposed to UV radiation. That comes from neural crest cells as well. So melanocyte production, these are ones that typically take a dorsolateral migration. These others, sensory neurons, Schwann cells, adrenal gland cells, these typically take a ventrolateral migration. So, I mean, think about it. Dorsolateral means they're going to be on the peripheral right next to the skin, and they're going to be part of the dermis, and they're going to form melanocytes in the dermis of your skin. Whereas if they migrate more ventrally or internally, they're typically going to become these other types of cells. So all of it comes down to paracrine signaling. As these neural crest cells are migrating through their respective areas, they're receiving the necessary paracrine signals to differentiate into sensory neurons or Schwann cells or adrenal cells or whatnot. Here's an example of a Schwann cell. The Schwann cells will wrap themselves around the axons. So the axons are the projections of the neurons that propagate the signal from your brain to your spinal column and your spinal column to your tissues. That's what allows your muscle to contract and your sweat glands to work and all of that. The myelination actually allows for the depolarization of your neurons to occur much more rapidly. You have fast neurons that will be able to propagate a signal in milliseconds. Those are usually myelinated. Then you have slow ones. All you guys know this. You get hit with a soccer ball in a particular area. You feel the pressure instantaneously. Those are the fast twitch ones. Those are the fast fibers. Then comes the pain, and those are the slow neurons that are not necessarily myelinated. Okay? So there is a difference between how fast some of these signals will propagate depending upon whether they're myelinated or not.